Hummingbirds and bees feast on the nectar of flowers. Prairie dogs nibble at and buffaloes graze on prairie grasses, while bears forage for berries and moose munch on the shoots of wetland vegetation. The goal of all of these organisms to take in the nutrients and chemical energy necessary to sustain life. Organisms like these that obtain their energy by consuming other living organisms are referred to as heterotrophs. On the other hand, plants are referred to as autotrophs. Autotrophs are organisms that are able to use a source of energy, such as sunlight, to produce food directly from simple inorganic molecules in the environment. Plants do this through a process called photosynthesis, in which they use light energy to convert carbon dioxide and water molecules into glucose and other carbohydrates in which they store energy. Plants, of course, don't deliberately store energy in carbohydrates like glucose for the use of herbivores, but rather for use in processes such as synthesizing flower pigments and growing roots, stems, branches, and trunks. But much of their stored energy is nonetheless consumed by animal predators who eat the leaves and stems of plants, often injuring or destroying them. Many plants, however, do deliberately make available large amounts of high-energy carbohydrates to animals in the nectar of their flowers and fruits to attract animals, which then unwittingly help the plant scatter its pollen or sow its seeds. In order for the energy stored in glucose or other carbohydrate molecules to be used by the plants themselves or by animals consuming the plant or its fruits and nectars, the energy has to be transferred to ATP molecules. ATP, or adenosine triphosphate molecules, are the universal currency of energy within both plant and animal cells. ATP molecules are formed as a result of a high-energy bond joining an inorganic phosphate group to an adenosine diphosphate, or ADP, molecule. Energy is stored in the bond between the phosphate group and the ADP molecule. ATP molecules store energy in a form that provides immediate availability for urgent cellular processes. The demand for ATP molecules by organisms performing work can be extremely high. For example, marathon and cross-country runners often break down a pound or more of ATP molecules in a minute. Of course, if they are to continue running, the ADP molecules and the free phosphate groups produced by the breakdown of ATP molecules have to be immediately recombined to form new ATP molecules. Two different processes can take the energy stored in glucose or another carbohydrate and form new high-energy ATP molecules for a runner: glycolysis and cellular respiration. Glycolysis can occur without the presence of oxygen and takes place in the cytosol, or the fluid found in the cytoplasm of cells. In we humans, glycolysis often provides the energy for events such as 100-meter dashes and weightlifting that require short bursts of energy. These types of activities depend on glycolysis because they happen so suddenly that the cells of the body cannot be oxygenated rapidly enough by the heart and lungs to support the second means of converting glucose into energy, cellular respiration, which requires oxygen. Glycolysis is not a very efficient process. It only releases approximately 2% of the energy stored in a molecule of glucose, which translates into about 2 ATP molecules produced for every glucose molecule metabolized. Fortunately, cellular respiration provides a much more efficient way of using the energy stored in the glucose. But as a distance runner's gasping for air suggests, the process of cellular respiration is highly dependent on the availability of oxygen. Cellular respiration converts approximately 37% of the energy stored in a glucose molecule into immediately usable energy in the form of between 36 and 38 ATP molecules. While this does not sound like a tremendous figure, it is more efficient than either automobile or jet engines are at converting their respective fuels into usable energy. Most cells can metabolize a variety of organic molecules other than glucose for energy, including more complex carbohydrates, proteins, and fats. But in this program, for a number of reasons, we will focus specifically on how cells convert glucose into energy. First, the cells of most multicellular organisms at some time or another burn or metabolize glucose and convert it into energy. And many types of cells, such as the nerve cells of the human brain, burn glucose nearly exclusively. Secondly, when organic molecules other than glucose are used in cellular respiration, they are usually broken down and converted into glucose or similar molecules before undergoing the process of cellular respiration. Finally, it is useful to visualize cellular respiration as a set of reactions that reverse the reactions involved in photosynthesis. And photosynthesis is usually explained in terms of synthesizing glucose molecules from carbon dioxide and water molecules in a process powered by light energy, even though other carbohydrates can be synthesized via photosynthesis. When we compare the equations for photosynthesis and complete glucose metabolism through cellular respiration, we notice that they are mirror images of one another. Photosynthesis starts with 6 molecules of carbon dioxide and 6 of H2O, adds light energy, and converts the reactants into a glucose molecule and 6 oxygen molecules. Cellular respiration, on the other hand, takes a glucose molecule and 6 molecules of oxygen and converts them back into 6 molecules of CO2 and 6 of H2O, releasing energy in the process. The process of breaking apart glucose molecules to obtain their energy always starts with glycolysis. What happens next depends on whether anaerobic or without oxygen conditions or aerobic with oxygen conditions exist within the cell. If anaerobic conditions exist within the cell, the next stage is fermentation. Fermentation does not produce any ATP energy, but it allows the process of glycolysis to repeat itself. If an aerobic situation exists within a cell, the next step is cellular respiration, in which oxygen is used to break down the component parts that result from glycolysis into carbon dioxide and water, while releasing substantial amounts of energy. Let's now look at each of these stages in more detail. Glycolysis literally means to break apart glucose. That is exactly what happens in the complex sequence of reactions which break down a molecule of glucose into 2 molecules of pyruvic acid. The process of glycolysis, which occurs in the cytosol of a cell, can be broken down into 2 major stages: glucose activation and energy harvest. The process of glucose activation uses the energy of 2 ATP molecules to convert glucose into highly reactive fructose diphosphate. Once a glucose molecule is converted to fructose diphosphate, energy harvesting can begin. At the beginning of energy harvesting, the fructose diphosphate molecule breaks apart into 2 molecules of phosphoglyceraldehyde, or PGAL for short. You will perhaps remember from your study of photosynthesis that 2 PGAL molecules are combined to form a glucose molecule in the process of photosynthesis. After the PGAL molecules are formed, they go through a series of reactions that end with the production of 2 molecules of pyruvic acid, one from each PGAL molecule. The reactions that transform PGAL molecules to pyruvic acid also generate 2 ATP molecules from each PGAL molecule converted to pyruvic acid, for a total of 4 ATPs for each original glucose molecule. Since 2 ATPs were used during glucose activation to convert a glucose molecule into fructose diphosphate, there is a net gain of only 2 ATPs per glucose molecule broken down during glycolysis. But the potential for forming more ATP exists as a result of the energy harvest. In addition to the ATP molecules formed during the conversion of PGAL molecules to pyruvic acid molecules, each PGAL molecule donates an electron and a hydrogen atom to an NAD+ molecule, converting it into an energized NADH molecule. The energy of the 2 NADH molecules formed during this process can be used to synthesize more ATP, but only where oxygen is available, allowing cellular respiration to occur. Before we look at cellular respiration, however, let's look at what happens when no oxygen is available. Under anaerobic conditions, the energy of the electrons carried by the NADH molecules are wasted, and valuable positively charged NAD+ molecules, which are necessary for getting electrons and hydrogen ions out of the way during the breakdown of glucose into pyruvic acid, are used up. If a cell runs out of positively charged NAD+ molecules, glycolysis stops. It is through the process of fermentation that NAD+ molecules are regenerated so that glycolysis can continue. There are 2 major types of fermentation: lactic acid fermentation and alcoholic fermentation. Lactic acid fermentation often occurs in humans during and after strenuous exercise. When you exercise, your muscles need lots of ATPs as an energy source. Even though cellular respiration generates much more ATPs than glycolysis alone, cellular respiration is limited by organismal respiration. That is, that you may not be able to get enough oxygen out of the air into your blood and delivered to your muscles to keep cellular respiration going at the necessary pace. But instead of stopping completely, the cells continue the process of glycolysis, which provides its meager 2% of the energy available in glucose molecules. As a result of this continued glycolysis, both pyruvic acid and NADH molecules pile up. In order to regenerate the NADH+ molecules necessary in order for glycolysis to continue, muscle cells convert pyruvic acid molecules to lactic acid, using the electrons and hydrogens from the NADH as an energy source. The restored NAD+ molecules are now free again to accept electrons during the process of glycolysis, and energy production can continue. There is one drawback, however. Lactic acid is toxic in high concentrations in cells. Eventually, muscles suffer from lactic acid poisoning and fatigue sets in. After exercise ceases, the built-up lactic acid is converted back to pyruvic acid, which, as we will see later, is broken down through cellular respiration to carbon dioxide and water. There are a variety of microorganisms, including certain bacteria, that free up NADH+ ions through lactic acid fermentation. These bacteria are often added to milk or milk products and dairies in order to produce dairy products such as yogurt, sour cream, and cheese via lactic acid fermentation. The second method of fermentation, used by many microorganisms, is alcoholic fermentation. This fermentation process, rather than producing lactic acid, produces ethanol, commonly referred to as alcohol, and CO2 from pyruvic acid, using hydrogen and electrons from NADH molecules. Alcohol fermentation, like lactic acid fermentation, can be a somewhat chancy proposition for an organism because alcohol, like lactic acid, is toxic above certain concentrations. Most yeast, for example, die if alcohol concentrations exceed 12%, although certain types of yeast have been developed for wine production that can exist in concentrations of alcohol up to 18%. Wines, of course, are produced as yeast break down the sugars of grapes or other fruits in an environment without oxygen. Sparkling wines, such as champagne, are bottled while the yeast are still alive and fermenting, thus trapping both alcohol and CO2 within the bottle. When an abundant supply of oxygen is available to a cell, fermentation does not go on at all, but rather cellular respiration occurs. Cellular respiration occurs not in the cytosol of the cell, but rather in organelles called mitochondria, which in some ways can be compared to the chloroplasts of plants. But rather than building glucose molecules, they break glucose molecules down into carbon dioxide and water. Mitochondria have two membranes, producing two compartments. An inner compartment, enclosed by the inner membrane and containing the mitochondrial matrix, and an intermembrane compartment between the inner and outer membranes. The first step of cellular respiration is glycolysis, which we looked at earlier. As you'll remember, glycolysis takes place in a cell's cytosol and produces 2 molecules of pyruvic acid per molecule of glucose. In order to proceed with the process of cellular respiration, the 2 pyruvic acid molecules produced by the division of each glucose molecule must be transported through both mitochondrial membranes into the mitochondrial matrix. Large pores in the outer membrane of the mitochondria make it highly permeable to pyruvic acid molecules. Pyruvic acid molecules cross the inner membrane through special proteins. Once within the matrix, each of the 2 pyruvic acid molecules is split into a CO2 molecule and a 2-carbon molecule called an acetyl group. Each acetyl group immediately attaches to a molecule called coenzyme A, forming an acetyl coenzyme A complex, or acetyl-CoA for short. During these reactions, an energetic electron and a hydrogen are transferred to positively charged NAD+ ions, forming one energy-carrying NADH molecule per acetyl-CoA. The 2 acetyl-CoA molecules enter a cyclic pathway known variously as the Krebs cycle, referring to its discoverer Hans Krebs, or the citric acid cycle, named for the first product in the reaction sequence, citric acid. We'll refer to it as the Krebs cycle. As the Krebs cycle begins, each of the 2 acetyl-CoAs combines briefly with a molecule of oxaloacetic acid. The 2 carbon acetyl groups are donated to the 4 carbon oxaloacetic acids to form 2 6-carbon citric acid molecules, while the coenzyme A's of each acetyl-CoA are released to be used again. Enzymes within the mitochondrial matrix then lead the 2 citric acid molecules through a number of rearrangements that regenerate the 2 oxaloacetic acid molecules that initially help form the citric acid molecules. During this process, 4 CO2 molecules are given off, and the energy of the 2 acetyl groups is used to form 2 ATP molecules and 8 electron carrier molecules, 2 FADH2 molecules, and 6 NADH molecules. In addition, you'll recall that earlier 2 NADH molecules were produced as the 2 acetyl-CoAs that entered the Krebs cycle were formed. At this point, you will notice that the cell has still only gained 4 ATP molecules from the original glucose molecule, 2 that were formed during glycolysis and the 2 formed during the citric acid cycle. However, glycolysis and the citric acid cycle have allowed the cell to capture many energetic electrons and attach them to carrier molecules. 2 NADH during glycolysis, 2 during acetyl-CoA formation, and 6 more during the Krebs cycle, along with the 2 FADH2 molecules also formed during the Krebs cycle, for a total of 10 NADH and 2 FADH2 molecules. Each one of these carrier molecules hold a pair of energetic electrons. These carrier molecules deposit their energetic electron pairs on electron transport systems embedded in the intermitochondrial membrane. The energetic electron pairs move from molecule to molecule along the electron transport system. Energy released by the electrons during these transfers from molecule to molecule is used to pump hydrogen ions from the mitochondrial matrix across the inner membrane and into the intermembrane compartment. As a result, the mitochondrial matrix becomes negatively charged, while the surrounding intermembrane compartment becomes positively charged. This creates a source of energy that will be used later as positively charged hydrogen ions flow back into the matrix through channel proteins due to their electrical attraction to the negatively charged mitochondrial matrix inside the inner membrane. Enzymes attached to the channel proteins use the energy created by the flow of the hydrogen ions to create high-energy ATP molecules by adding phosphate-based molecules to ADP molecules. Finally, at the end of the electron transport system, enzymes take electrons coming off the electron transport system and combine them with oxygen atoms and positive hydrogen ions to form water. This clears out the transport system, leaving it ready to run more electrons through. Without oxygen, the electrons pile up in the transport system, with the result that hydrogen ions would no longer be pumped across the inner membrane into the intermembrane compartment. The flow of hydrogen ions through the ATP synthesizing enzymes produced by each molecule of glucose broken down provides the energy to synthesize 32 to 34 molecules of ATP by combining 32 to 34 ADP to phosphate groups in the matrix. Another way of looking at it is that, on average, each pair of electrons provided by either an NADH or FADH2 molecule traveling down an electron transport system pumps enough hydrogen ions across the inner membrane to provide the energy for the production of 3 ATP molecules. The final step in cellular respiration is ATP transport. The outer mitochondrial membrane is very permeable to ATP, ADP, and inorganic phosphate molecules. Proteins in the inner mitochondrial membrane exchange ATP from the matrix for ADP and phosphate groups, which have diffused into the intermembrane compartment through the outer mitochondrial membrane. The ATP molecules, now out in the intermembrane compartment, eventually diffuse across the outer mitochondrial membrane into the cell cytoplasm, where they provide most of the energy needed by a cell for activities such as building protein molecules, moving organelles throughout the cytoplasm, and in humans, providing the energy for the expansion and contraction of muscle cells necessary for running, jumping, and all other types of motion. As we have seen, the complete breakdown of 2 pyruvic acid molecules into carbon dioxide and water in the mitochondria results in an impressive gain in ATP molecules for the cell. Glycolysis by itself produces only 2 molecules per glucose molecule, but the reactions in the mitochondria add 34 to 36 more ATPs for a total of 36 to 38 ATPs per glucose molecule. Today, most organisms are so dependent on the additional ATPs produced by cellular respiration that anything that interferes with cellular respiration, such as a lack of oxygen due to suffocation or stopping of the heart, quickly results in the death of the organism. But as we saw earlier, this wasn't always true. It wasn't until prokaryotic cells capable of photosynthesis started releasing large amounts of oxygen into the atmosphere, and other prokaryotes capable of using that oxygen to carry out cellular respiration took up residence in larger eukaryotic cells, that the stage was set for large multicellular organisms to develop. The increased energy made available through cellular respiration was essential for the development of nearly all modern species, including we humans, who depend on tremendous amounts of energy, not only to power our physical activities, but also to power our mental processes that allow us to not only exist here on Earth, but to thrive and explore not just the Earth, but the far reaches of the universe.